Alright. Well, we now move forward to Krebs cycle. We now know that we got pyruvate from glucose, also we get pyruvate from amino acids and from galactose and fructose. This pyruvate in-linked reaction makes acetyl-CoA. Acetyl-CoA in addition also comes by the oxidation of ketones, fatty acids and alcohol. Now this acetyl-CoA enters into the Krebs cycle, which is the most important cycle, most important pathway in entire metabolism. Now this acetyl-CoA makes citrate, then isocitrate, alpha-ketoglutarate, then succinyl-CoA, succinate, fumarate, malate, oxaloacetate, and then the citrate. This keeps on repeating. And here the important thing is that dehydrogenases. And now you know what are dehydrogenases. Dehydrogenases, whenever the word dehydrogenase comes, NAD has to be reduced to NADH. So this is the first NADH we get in the Krebs cycle. This is the second NADH, so there will be dehydrogenase. Yes, there is dehydrogenase, alpha ketoglutarate dehydrogenase, and then there is another FADH2. There should be a dehydrogenase, and the dehydrogenase is succinate dehydrogenase, and then malate dehydrogenase. So there should be NADH, and this is the third NADH. First NADH, second NADH, and third NADH. So actually we get 3 NADH. One molecule of acetyl-CoA enters into the Krebs cycle. In addition, we get 1 FADH2. Okay. Now you see that there is one carbon dioxide here and the second carbon dioxide here. So this is the acetyl-CoA carbon dioxide. This is the carbons of acetyl-CoA. The first carbon dioxide and the second carbon dioxide. So the rate limiting step for this pathway is isocitrate dehydrogenase. If isocitrate dehydrogenase is active, is fast, the cycle will be fast, and if it is slow, the cycle will be slow. So let's see which things make it fast. If there is less NADH, if there is less NADH in the cell, if there is less FADH2 in the cell, and if there is less ATP in the cells, isocitrate dehydrogenase will be fast. And if there is more AMP in the cell, the isocitrate dehydrogenase will be fast. But if these things are high, if NADH, FADH2, ATP and ATP are high while AMP is low, and NADH, FADH2 and ATP are high, then isocitrate dehydrogenase slows down. And once this slows down, citrate starts to accumulate. And if citrate starts to accumulate, this citrate leaves the mitochondria and enters into the cytoplasm for fatty acid synthesis and cholesterol synthesis. And cholesterol synthesis via the citrate shutdown. This is an important point. If isocitrate dehydrogenase is slow because of excess of NADH, FADH2 and ATP, then the citrate starts to accumulate and that citrate leaves the cell to make fatty acid and cholesterol. Right? And if you remember from the overview of energy metabolism, the first lecture, all that acetyl-CoA starts making fatty acid and cholesterol when the energy needs of the cells are met. All right. Another important point is that this succinyl-CoA, which is produced in Krebs cycle, this is important for making heme. Heme is not only important for hemoglobin and myoglobin, but it is also important for making cytochromes. And cytochromes are very much important in electron transport chain as well as in the liver metabolism. In addition, heme is also an important component of catalase and peroxidase, which are antioxidants, which acts as antioxidants, catalase and peroxidase. Right? This is an important point. Succinyl-CoA makes heme, and heme is not only important in hemoglobin and myoglobin, it also is important for cytochromes and catalase and peroxidase. And then fumarate. You see that this fumarate, this can link with urea cycle. Urea cycle produces fumarate, and then this fumarate becomes the part of the Krebs cycle. Right? Another important point is that this malate leaves the cell and is important in gluconeogenesis. We will see that in a while. These are the important points. You see that this is actually the Krebs cycle. Krebs cycle is important for fatty acid synthesis. When the Krebs cycle is slow, the fatty acid and cholesterol synthesis happens from citrate, and also there is the heme formation from succinyl-CoA in the Krebs cycle. So now we see the electron transport chain. It is really very easy. You see that all those pathways like link reaction, like Krebs cycle, like beta oxidation of fatty acids, or the amino acid oxidation, or the ketone body oxidation, or alcohol oxidation, they produced NADH. Now these NADH gives their electrons to electron transport chain, gives their electron to electron transport chain. This is taken by NADH dehydrogenase complex 1, and now these electrons are handed over to coenzyme Q. Now coenzyme Q also gets electron from glycerol phosphate shuttle. And if you remember, glycerol phosphate was produced in glycolysis and was important in transferring the electrons from glycolysis. So this glycerol phosphate shuttle, FADH2, gives its electron to coenzyme Q. Similarly, succinate dehydrogenase complex 2, which is also the part of the Krebs cycle, gives its electron to coenzyme Q, and then fatty acid oxidation produces FADH2, and those FADH2 are taken up by the coenzyme Q. Now the coenzyme Q hands over their electrons to cytochrome B by C1 or complex 3, and complex 3 hands over electrons to cytochrome C, and cytochrome C hands over the electron to cytochrome A by A3 or the complex 4, and finally these electrons are accepted by oxygen. They are gained by oxygen, and oxygen is reduced to water. And now, and you know that electrons move, current is produced, electricity is produced, energy is produced. That energy is sufficient to push these protons from the matrix, into the intermembrane space, You see that this inner membrane, this is the intermembrane space, and now in the intermembrane space there are a lot of protons. There are a lot of protons accumulated because of the movement of electrons in the electron transport chain. I again say that when electrons are transported in the electron transport chain, current is produced. That current, that energy is sufficient to push the protons from the mitochondria into the intermembrane space. And once these protons are accumulated over here, they move back to mitochondrial matrix. By F0 F1 complex, and when they move back to the mitochondrial matrix, again the charges are moving, so the energy is produced. That energy is sufficient to convert this ADP into ATP, and then that ATP moves out of the cell by ATP-ADP translocase, and it is used in different energy purposes. All right, so this is the whole story, right? So if make it a little zoom in. If I zoom in, right, so you see that so this is the whole story. Electrons from NADH and FADH2 are handed over to the electron transport chain. In the electron transport chain, these electrons move, and when these electrons move in the electron transport chain, energy is produced. That energy pushes the protons from the mitochondrial matrix into the intermembrane space, and when these protons move back to the mitochondrial matrix, energy is produced, and that energy converts ATP into ATP, and that ATP is used in different cell processes. But sometimes, because of the presence of uncoupler proteins, which are aspirin in high doses thermogenin, these protons do not couple with F0 F1 complex. Rather they move directly from the intermembrane space back to the mitochondrial matrix, and in that process energy is produced, and that energy is lost in the form of heat instead of ATP. Another important point, the complex 4 of the electron transport chain is inhibited by cyanide and carbon monoxide. Complex 5, it's also called complex 5, F0 F1 complex, is inhibited by oligomycin, while complex 1 is inhibited by barbiturates and rotenone. Well, rotenone is an insecticide, and barbiturates, they are used to depress the central nervous system, right? And they are also used as anesthetics, as an adjunct in anesthetics. So this is electron transport chain. All right. So now we move to glycogenesis. We know that this glucose is entering into the liver. All right. That glucose makes glucose 6-phosphate first in the glycolysis. Right. Now from the glucose 6-phosphate, it starts making glucose 1-phosphate, and then that glucose 1-phosphate makes UDP glucose. and then that UDP glucose by the enzyme glycogen synthase and branching enzyme makes glycogen. So this was one step. The glucose 6-phosphate starts making glucose 1-phosphate, and then glucose 1-phosphates through UDP glucose starts making glycogen. And glycogen synthesis happens in the presence of insulin. You know, all synthesis happens under the influence of insulin. Right, and insulin actually activates glycogen synthase, and glycogen synthase starts making glycogen. This glycogen is stored in the liver. But in the glycogenolysis, when we need glucose during fasting or starving, there are the hormones like epinephrine, like glucagon. They now activate glycogen phosphorylase and debranching enzyme. Glycogen phosphorylase and similarly AMP in the muscles. When there is less energy in the AMP is actually the indicator of the energy. I mean, if there is less energy, there will be more AMP, and if there is more energy, there will be a lot of ATP. So now this AMP or the epinephrine or glucagon and even cortisol as well, right? So these hormones activate glycogen phosphorylase and debranching enzyme, and glycogen is broken down to glucose 1-phosphate. And now this glucose 1-phosphates make glucose 6-phosphate, and that glucose 6-phosphate can be used in glycolysis to make pyruvate. But in the starvation, we need glucose in the blood. So this glucose 6-phosphate makes glucose, and this was the irreversible step of the glycolysis, which has now been reversed by the enzyme glucose 6-phosphatase. And this is an important enzyme of gluconeogenesis, actually the last enzyme of the gluconeogenesis we will see. So glucose 6-phosphatase actually converts glucose 6-phosphate back to glucose, and now glucose is free to leak out of the cell and enter into the blood, right? So glycogen breakdown produces glucose, and that glucose increases glucose level. Well, this concludes major portion of carbohydrate metabolism. Only two topics are left. One is gluconeogenesis, and the other one is hexose monophosphate shunt or the pentose phosphate pathway. I will be making a video on those two topics soon. Please leave your feedback, and if you have any question regarding this video, you can post them in comment field. Thank you so much. Hope to see you soon. Thank you. Bye bye.